Okay, so let's dive into this world that's usually hidden from view. The microscopic realm of chemicals in our waterways. Specifically, we're heading to Australia to uncover a bit of a mystery. How certain synthetic hormones, the kind found in oral contraceptives, are showing up in places they really shouldn't be.
Yeah, it's fascinating, isn't it? These compounds, they're designed for human use, yet they have these unintended consequences for wildlife in our rivers.
Exactly. And today we're taking a deep dive into a 2017 scientific paper. It was published in the journal Ecotoxicology and Environmental Safety
I think.
by Chatchaporn, Uraipong, and colleagues, Robin D. Allan, Chunhua Lee, Ivan R. Kennedy, Victor Wong, and Nanju Alice Lee. And it really sheds light on this whole issue.
Yeah, their work is quite significant. They focused on the Hawkesbury River in New South Wales, investigating the presence of two specific hormones, 17α-ethinylestradiol, or EE2, and mestranol, also known as ME-E2.
Now I know you're thinking, hormones in the water? How does that even happen?
Yeah.
Well, one way is through wastewater treatment plants. They do a great job of removing most pollutants,
Yeah,
but these hormones, they can kind of slip through the cracks, end up in rivers and streams.
that's right. And what's particularly interesting about Me-Too is that it actually converts into EE2 in the body. So we're essentially dealing with two sources of the same potent hormone.
Oh, wow. And here's where it gets really concerning. These hormones are known as endocrine-disrupting compounds, or EDCs,
Right.
and they can mimic or disrupt the natural hormones in both humans and wildlife.
Exactly. And even at very low concentrations, these EDCs can have significant impacts on aquatic life. They can interfere with fish development, reproduction, even their behavior. It's like throwing a wrench into this delicate machinery of an ecosystem.
So how did these researchers even go about detecting these hormones in the water? It's not like you can just dip a litmus paper in and get a reading.
Well, that's where the ingenuity of this study comes in. Uraipong and his team, they developed a new method for detecting EE2 and Me2 using a technique called an ELISA, or ELISAR, which stands for enzyme-linked immunosorbent assay.
Lisa, okay, you're going to have to break that down for me.
Yeah.
What is it and how does it work?
So think of it like a molecular lock and key system. ELISA utilizes antibodies, which are like highly specific locks that can only bind to certain target molecules. In this case, EE2 and ME2. The keys,
So these antibodies are like tiny detectives sniffing out these hormones in the water.
that's a great way to put it. The researchers actually designed these antibodies to be incredibly sensitive and specific to EE2 and Me2,
Okay,
so they wouldn't get confused by other similar molecules in the water.
that's pretty impressive. But how do you actually measure the amount of hormones present? Do the antibodies like light up or something?
It's actually a bit more complicated than that. The antibodies are attached to an enzyme that produces a color change when it binds to the target hormone. The intensity of the color change is directly proportional to the concentration of the hormone in the sample.
Ah, so it's like a molecular color chart.
Exactly.
The more intense the color, the more hormones are present. That's clever.
Exactly. And the beauty of ELISA is that it's relatively inexpensive and can be used to analyze a large number of samples quickly. It's a powerful tool for environmental monitoring.
So armed with their ELISA, the team set out to investigate two areas in New South Wales. They chose South Creek, a more urbanized area near Sydney, and Emigrant Creek, a more rural location in northern New South Wales. It sounds like they were trying to compare how human activity might be impacting hormone levels.
That's right. South Creek, being closer to Sydney, would likely have more wastewater treatment plants, other potential sources of these hormones.
Right.
Emigrant Creek, on the other hand, represented a less developed area, perhaps with less direct human impact.
Okay, so let's get to the results. What did they find? Were these hormones lurking in the Hawkesbury River?
Well, the results were quite revealing. They found detectable levels of EE2 and ME2 in both locations.
Wow, even in the more rural setting of Emigrant Creek. That's a bit concerning, isn't it?
It is. It suggests that these hormones are more widespread in the environment than we might initially think. They could be transported through groundwater or runoff from agricultural areas. But what's even more striking is the difference in concentrations between the two locations.
Okay, tell me more. How did the levels in South Creek compare to those in Emigrant Creek?
In South Creek, the EE2 and ME2 levels range from 15 to 29 nanograms per liter.
Okay.
And get this, those levels were especially high in samples taken downstream from sewage treatment plants.
So it seems like those treatment plants are a major source of these hormones in the river.
Right.
That makes sense, but it's still a bit alarming, isn't it?
Absolutely. And it really highlights the need for better wastewater treatment technologies or policies to address this issue. Even if these hormones are slipping through in small amounts, they can still have a significant impact on the ecosystem.
Now, what about Immigrant Creek? What were the levels like there?
The concentrations in Immigrant Creek were lower, ranging from 4.1 to 8.3 nanograms per liter.
Still detectable, though, which really reinforces your point about these hormones being more widespread than we might realize.
Exactly. It's a reminder that even in areas that seem relatively pristine, there's still potential for contamination. And it begs the question, if these hormones are present in the Hawkesbury River, are they present in other Australian waterways as well?
That's a great question, and one that I'm sure we'll explore further in this deep dive. Now, before we move on, I want to go back to the ELISA method for a moment.
Yeah.
You mentioned that it was relatively inexpensive and quick, but how do we know it's actually accurate? Did the researchers do anything to validate their results?
They did. They compared their ELISA results to those obtained using a more established and highly sensitive method. It's called gas chromatography-mass spectrometry, or GCMS. It's often considered the gold standard for this type of analysis.
So they were essentially double-checking their work using a more sophisticated technique.
Right.
And what did they find? Did the ELISA hold up?
The ELISA results align very well with the GCMS data,
Okay.
which gives us confidence in their findings. It demonstrates that this new ELISA method is a reliable tool for monitoring these hormones in the environment.
That's reassuring to know. Now, I understand that developing and using a new method like ELISA, it isn't always straightforward.
Right.
Did the researchers encounter any challenges along the way?
They did. Environmental samples are complex, and there are always factors that can interfere with the accuracy of a test. In this case, they found that things like humic acid, salinity, and pH levels in the water could affect the ELISA's performance.
Humic acid, salinity. Okay, we're getting a little technical here. Can you explain what those are and how they might interfere with the ELISA?
Of course. Humic acid is a complex organic molecule found in soil and water.
Okay.
It's a bit like a sticky web that can trap the target hormones,
Yeah.
making them less accessible to the antibodies in the ELISA.
So it's like the humic acid is hogging all the EE2, preventing the ELISA from getting a clear reading.
Exactly. And high salinity, which refers to the amount of dissolved salts in the water, can also affect the ELISA sensitivity. The salt ions can interfere with the binding between the antibodies and the hormones.
And what about pH? I know that's a measure of how acidic or basic something is.
Right.
How does that come into play?
Well, antibodies are quite sensitive to changes in pH. They work best within a specific pH range. If the water sample is too acidic or too basic, the antibodies might not function properly,
Yeah.
leading to inaccurate results.
So basically, these interferences highlight the need to carefully control the conditions when using ELISA for environmental analysis. It's not as simple as just dipping a test strip into the water.
That's right. But the researchers were aware of these potential interferences and took steps to address them. They adjusted the pH of the water samples and used a purification technique to remove humic acid before running the ELISA. It's a testament to their rigorous scientific approach.
Okay, so they tackled the challenges, validated their method, and uncovered some pretty significant findings. What does all of this mean? What are the broader implications of this study?
Well, for starters, it highlights the need for regular monitoring of these hormones in Australian waterways. We need to understand how widespread this contamination is and how it might be changing over time.
And I imagine it also raises questions about whether Australia should implement stricter regulations for these compounds like some other countries have done.
Absolutely. This study provides valuable data that can inform policy decisions. If we know that these hormones are present in our waterways at levels that could potentially harm aquatic life, then we need to consider what steps we can take to minimize their release into the environment.
And that brings us to the big question.
Yeah.
What are the potential effects of these hormones on fish and other aquatic organisms? We've hinted at it, but I think it's time to dive deeper into that topic.
You know, what's really interesting is that these hormones, EE2 and ME2, they belong to a group of molecules called steroidal sex hormones. And these molecules, they're notoriously tricky to differentiate because they're structurally very similar.
So it's like trying to tell identical twins apart.
Yeah.
How did the researchers make sure their ELISA could specifically detect EE2 and Me2 without getting confused by other similar hormones?
That's where the clever design of their ELISA comes in. They focused on a key component of ELISA called a hapten.
Hapten. Refresh my memory. What's a hapten again?
It's basically a small molecule that, when attached to a larger protein, it triggers an immune response. Think of it as a red flag that alerts the immune system to the presence of a specific target molecule.
So the hapten is like a wanted poster for EE2 and Me2.
Exactly. Now, in most EE2 ELISA tests, the linker that attaches the hapten to the protein is usually at a specific position on the EE2 molecule. It's called the C6 position.
Okay, so they attach this wanted poster to the E2 molecule. What's so special about that?
Well, Uraipong and his team, they did something different. They attached the linker at the C3 position instead.
Why would they do that? What's the advantage of attaching it at C3 instead of C6?
Here's the ingenious part. By changing the linker position, they essentially forced the antibodies to focus on a specific region of the EE2 molecule. It's called ring D.
Ring D. Why is that so important?
Ring D is the region that varies the most between different steroidal sex hormones.
Wow.
The other parts of these molecules are pretty much identical. So by targeting the antibodies to ring D, they made the ELISA much more specific to EE2 and Me2.
Ah, I see. It's like giving the antibodies a magnifying glass to zoom in on the unique features of EE2 and Me2, ignoring the common features shared by other hormones.
Precisely. It's all about enhancing the specificity of the test. And the results were impressive. Their ELISA showed very minimal cross-reactivity with other steroidal sex hormones.
That's great. So we can be pretty confident that the ELISA was accurately detecting EE2 and Me2 without getting fooled by other similar molecules.
Exactly. It's a testament to the power of thoughtful hapten design in immunoassay development.
Okay, let's shift gears a bit and talk about those interferences we discussed earlier. Things like humic acid, salinity, and pH. You mentioned that the researchers had to take steps to address these challenges.
Right.
Can you elaborate on that?
Of course. So they found that humic acid concentrations above a certain threshold, it could start to interfere with the ELISA. It's like the humic acid was acting like a sponge, soaking up the EE2 and preventing it from binding to the antibodies.
So it's almost as if the humic acid was masking the presence of the hormones, making it harder for the ELISA to detect them accurately.
Exactly. And they observed that salinity levels above a certain point could also affect the sensitivity of the ELISA. The salt ions in the water can interfere with the delicate binding interactions between the antibodies and the hormones.
It sounds like even slight changes in the water's chemistry could throw off the ELISA's accuracy.
That's right. And pH was another important factor. Remember, antibodies are proteins, and they're very sensitive to changes in pH. They function optimally within a specific pH range.
So if the water sample is too acidic or too basic, the antibodies might not work as effectively.
Exactly. And that could lead to inaccurate results. The researchers found that significant deviations from a neutral pH could significantly impact the ELISA's performance.
So how did they deal with these interferences? Did they just throw up their hands and say, well, this water is too messy to analyze?
Not at all. They came up with some clever solutions. For example, they adjusted the pH of the water samples before analysis to ensure it was within the optimal range for the antibodies.
So they essentially pre-treated the water samples to create the ideal conditions for the ELISA.
Precisely. And for samples with high levels of humic acid or salinity, they used a technique called solid phase extraction to purify the samples before running the ELISA.
Solid phase extraction, what's that?
It's a technique that uses a special material to selectively bind and concentrate the target molecules. In this case, EE2 and Me2. It's like using a magnet to pull needles out of a haystack.
So it's a way to remove the interferences and enrich the sample with the hormones they're interested in. That makes sense.
Exactly. By taking these steps, the researchers minimized the impact of these interferences and improved the accuracy of their results.
Okay, so we've talked a lot about the ELISA method, but let's not forget about the bigger picture. We're talking about hormones in our waterways. These are chemicals that can have profound effects on aquatic life. What did the researchers find in terms of the levels of EE2 and Me2 in the Hawkesbury River?
Their findings were quite striking, actually. In South Creek, the more urbanized area, they found EE2 and Me2 concentrations ranging from 15 to 29 nanograms per liter.
And those levels were particularly high downstream from sewage treatment plants, right? Which points to those plants as a major source of these hormones in the river.
Exactly. And while the levels in Immigrant Creek, the more rural site, were lower, ranging from 4.1 to 8.3 nanograms per liter, the fact that they were detectable at all is concerning.
It really drives home the point that these hormones are more pervasive in our environment than we might think.
Absolutely. And it underscores the need for continued monitoring and potentially stricter regulations to protect our waterways.
Now, I remember reading that the researchers were able to achieve an even lower detection limit with their ELISA. Can you tell me more about that?
Yes, you're right. When they combined the ELISA with that solid phase extraction technique we talked about earlier, they were able to detect EE2 and MeE2 at concentrations as low as zero point zero 0.05 nanograms per liter.
Wow, that's a thousand-fold improvement. That's incredible.
It is. It shows how powerful these analytical techniques can be when used in combination.
So we've got this sensitive and specific ELISA method. What does this mean for future research and monitoring efforts?
Well, this ELISA method, it offers a valuable tool for researchers and environmental agencies to monitor these hormones in various water sources. Its sensitivity allows for the detection of even trace amounts of EE2 and MeE2.
And its specificity ensures that they're accurately measuring these specific hormones without interference from other similar compounds.
Precisely. This method could be used to assess the effectiveness of wastewater treatment processes, track the fate of these hormones in the environment, and evaluate the potential risks to aquatic ecosystems.
It's like having a new weapon in our arsenal to combat this invisible threat.
Exactly. It's a significant step forward in our understanding and ability to address the issue of endocrine disrupting compounds in our environment.
Okay, I think we've covered the technical aspects of this study quite thoroughly. Now I want to circle back to the bigger picture, the potential impacts of these hormones on aquatic life. We've touched on it briefly, but I think it deserves further exploration. We've been talking about these synthetic hormones, EE2 and MeE2, showing up in the Hawkesbury River, but why should we care? What's the big deal about a few nanograms per liter of these compounds in the water?
Well, the concern lies in their ability to disrupt the endocrine system, which is essentially the body's hormonal communication network. And even tiny amounts of these hormones can mimic or block the actions of natural hormones, throwing off these delicate physiological processes.
So it's like sending mixed signals within an organism's body.
Precisely. And in the case of fish, these mixed signals can have a range of, concerning effects. For instance, studies have shown that exposure to EE2 can lead to the feminization of male fish.
Feminization, what does that mean exactly?
It means that male fish start developing female characteristics. They might develop eggs in their tests, exhibit altered mating behaviors, or even experience reduced sperm production.
Wow, that's pretty dramatic. It sounds like these hormones are essentially hijacking the fish's reproductive systems.
That's a good way to put it. And these effects aren't limited to just a few individuals. If enough fish in a population are affected, it can skew the sex ratios, leading to a decline in overall reproductive success.
So it's not just about the individual fish, it's about the health of the entire population and ultimately the ecosystem.
Exactly. These endocrine disrupting compounds can have cascading effects throughout the food web. A decline in fish populations can impact predators that rely on them for food, as well as the smaller organisms that fish prey upon.
It's a domino effect, rippling through the entire ecosystem. And to think it all starts with these seemingly insignificant traces of hormones in the water.
It's a stark reminder that even small changes at the molecular level can have profound impacts on the environment.
Now, the researchers in the study focused on EE2 and MeE2, but are there other endocrine-disrupting compounds that we should be concerned about?
Absolutely. EE2 and Me2 are just the tip of the iceberg. There are many other synthetic and natural compounds that can interfere with hormonal systems. These include pesticides, industrial chemicals, and even some pharmaceuticals.
So this is a much broader issue than just hormones from contraceptives.
Definitely, endocrine disruption is a complex and pervasive problem that requires a multifaceted approach to address.
The researchers in this study suggested that Australia might need to consider stricter regulations on these compounds. What kinds of regulations are we talking about?
Well, one approach is to set limits on the allowable concentrations of these compounds in wastewater discharge. This would require upgrading wastewater treatment plants to remove these compounds more effectively.
So we're talking about investing in better technology to filter out these harmful substances before they reach our rivers and streams.
Exactly. Another approach is to regulate the use and disposal of products that contain these compounds. For example, some countries have banned the use of certain pesticides known to have endocrine disrupting effects.
It sounds like a combination of technological advancements and policy changes is needed to tackle this problem effectively.
Absolutely, and public awareness is also crucial. People need to understand the potential impacts of these compounds and make informed choices about the products they use and how they dispose of them.
So what can we as individuals do to minimize our contribution to this problem?
Well, there are several things we can do. First and foremost, dispose of unused medications properly. Never flush them down the toilet or throw them in the trash. Instead, look for drug take-back programs in your community.
That makes sense. Those medications can end up in our waterways
Yeah.
if they're not disposed of correctly. What else can we do?
Be mindful of the household products you use. Many cleaning agents, personal care products, and even some plastics contain endocrine-disrupting compounds. Look for eco-friendly alternatives and choose products with minimal packaging.
It's about making informed choices and considering the potential impacts on the environment.
Exactly. And support organizations that advocate for water protection and stricter regulations on endocrine-disrupting compounds.
We can use our voices and our actions to push for positive change.
Absolutely. Every little bit helps.
Well, I think this has been an incredibly insightful deep dive. We've explored the complexities of endocrine disruption, learned about a new method for detecting hormones in water, and discussed the implications for aquatic ecosystems and human health.
It's been a fascinating discussion, and I hope it encourages listeners to think critically about the hidden chemicals in our environment and the steps we can take to protect our precious water resources.
Thank you for joining us on this journey into the world of endocrine disrupting compounds. Remember, knowledge is power, and by understanding the challenges, we can empower ourselves to make a difference.